Hello, welcome to glycolysis. It's yet another interesting topic to discuss. Glycolysis comes from two words, that is glycos, meaning sugar, and lysis, meaning dissolution or bleach. So basically, glycolysis means split splitting of sugar, where that sugar is glucose, to give us energy which is required in the living cells. The most comprehensive pathway is the Embden-Messer pathway, which represents the complete steps of glycolysis in living cells. We start with glucose as the substrate and end up with pyruvate as the end product, where the reaction taking place in presence of oxygen, or lactic acid in absence of oxygen. In organisms that perform these cellular reactions, it takes place in the cytosol or in the cytoplasm, as it is known, and it's broken down into three stages. We have the energy requiring phase, which uses ATP to perform its function. We have the splitting phase, where now the 6-carbon compound is broken down into 2 3-carbon compounds. We have the energy releasing phase, where ATP now is produced. The reverse of glycolysis is known as gluconeogenesis, which is the generation of glucose from non-glucose sources. Now this chart here represents all the 10 steps of glycolysis, where in the energy requiring phase, ATP is used in step 1 and step 3 to give, for example, in step 1, in the presence of the hexokinase, which is an enzyme, will give us glucose 6-phosphate. So the compound is now bearing the phosphate groups. And now in the splitting phase, you can see fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and a glyceraldehyde 2-phosphate. This is the splitting phase. In the energy releasing phase, we have the phosphoglycerate kinase enzyme, which catalyzes the reaction of 1,3-bisphosphoglycerate, which are 2 molecules, to give us 3-phosphoglycerate, 2 molecules of them, to give us the 2 ATPs. And here in the last one, phosphoenol pyruvate is converted into pyruvate in presence of ADP to give us ATP molecule, which is the energy currents now we are talking about. So, like we have talked from the chart, we have the energy requiring phase, the energy splitting, the molecule splitting phase, the energy releasing phase. Now, for example, in the energy releasing phase, the 3-carbon sugar is converted into pyruvate. Pyruvate has some other functions which it performs in other pathways in the body, as we are going to see in our upcoming lectures. Now, the net production, we will have two ATPs which are produced and one NADH is produced per molecule. So the overall energy produced will be two adenosine triphosphate molecules and the two energy carriers, which are NADH. Overall speaking, glycolysis converts 6 carbon molecules, that's glucose, into 3 carbon molecules of pyruvate. The net products of this process are 2 molecules of ATP and 2 molecules of NADH. NADH, of course, comes from nicotinamide, which is a very important vitamin B complex, that is vitamin B3. Now in this slide, as you can see, we have the structure representation of glucose molecule and what happens. For example, here in step 1, glucose is phosphorylated at carbon number 6, making glucose to become glucose 6-phosphate. This is the phosphorylation stage, which is the initial stage. If you can go through this reaction, this pathway, you can be able to see the additions which are made in these steps. Again, in step 3, there is phosphorylation, which will happen at carbon number 1, now making it fructose 1,6-bisphosphate. The following now stage is the splitting stage, where we will be having 2 molecules which are produced from fructose 1,6-bisphosphate by the use of fructose 1,6-bisphosphate aldolase. Now, in this slide, we have the breakdown of the 10 stages of glycolysis, but in this stage, in this slide, we have the 3 steps, which is step number 1. Phosphate group is transferred to the glucose to make a glucose 6-phosphate. In step 2, glucose 6-phosphate is converted into its isomer, which is fructose 6-phosphate. Remember from chemistry, isomers are chemical compounds with the same molecular formula but different arrangements. So the molecular formula of glucose 6-phosphate and fructose 6-phosphate is the same, but with the help of the isomerase enzyme, it will be converted into fructose 6-phosphate. In stage 3, now energy in form of ATP is used to give us fructose 1,6-bisphosphate. Now in stage 4, fructose 1,6-bisphosphate is split into 2,3-carbon sugars, like we said before. That is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These two, three carbon compound molecules are simply isomers. In stage 5, we will have the 2-dihydroxyacetone phosphate, which is converted into glyceraldehyde 3-phosphate. Now the two molecules obviously exist in an equilibrium form. Now, this is the representation of stage 6 to 10, where the 3 carbon molecules are converted into 1,3-bisphosphoglycerate with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. In the next, we have a production of 2 ATP molecules in the presence of phosphoglycerate kinase. From there, we will form 3 phosphoglycerates. The 3 phosphoglycerates, in the presence of enzyme phosphoglycerate mutase, will mutate into 2 phosphoglycerates. The 2 phosphoglycerates, in the presence of enzyme enolase, will produce water and phosphoenol pyruvate. Now, phosphoenol pyruvate, in the presence of pyruvate kinase, will give us now the final product, which is pyruvate. The literature in this slide is used to explain what we have seen from the chart from the previous slide. Again, at this stage, this is the final stage where phosphoenol pyruvate donates phosphate to ADP, which is adenosine diphosphate, making it adenosine triphosphate. So the end product will be pyruvate. That's when it is in the presence of oxygen.